In this video, we'll explore viruses, tiny infectious particles that blur the boundaries of life. Viruses can replicate and evolve, yet they depend entirely on living cells to carry out these processes. We will look at the similarities and differences in virus structure, the lytic and lysogenic reproductive cycles, the diversity in virus structure, and theories around the origin and evolution of viruses. These are some of the most important key concepts covered in the IB biology syllabus. Despite viruses' wide diversity, they still share a basic structure. Viruses are universally small, between 20 and 300 nanometers in diameter, which allows them to enter their host cells. Because they don't have their own organelles or metabolism, they can achieve these small sizes that even bacteria cannot. Because they do not contain their own organelles, they are obligate parasites, meaning they must use other cells in order to complete their life cycles. Viruses remain a fixed size because they do not grow and develop, which is one of the key characteristics of life. So their size does not change. Viruses use the organelles and metabolism of their hosts for reproduction, showing they do not meet this characteristic of life. For example, they will use the host cell's ribosomes to assemble viral proteins, and the host cell's nucleotides to synthesize viral nucleic acids. Thus, they are considered an obligate parasite, or a parasite that is completely dependent on its hosts for survival and reproduction. Each virus consists of a core of genetic material, which can be either DNA or RNA, surrounded by a protective protein coat called a capsid. The capsid is made of repeating protein units that can self-assemble and give viruses a symmetrical shape. Viruses do not contain cytoplasm, and if they contain enzymes, only a small amount of them are contained in the capsid, as viruses use the host enzymes to replicate. Despite the similarities viruses share, they also display large variations in their structures. Some viruses are enveloped, meaning they have an additional outer layer called an envelope, which is made from the phospholipids of the host cell membrane and contains viral glycoproteins that help the virus attach to new host cells. These viruses tend to infect animal cells. Other viruses lack this membrane and are described as non-enveloped. They do not have a membrane that surrounds it, and they tend to infect bacteria and plant cells. The variety of virus structures reflect the wide range of strategies viruses use to infect their hosts. Viruses replicate by taking control of the host cell's machinery to make copies of themselves. When viruses infect a host, they insert their viral genetic material in order to use the host's ribosomes and metabolism to reproduce viral genetics and viral proteins. The genetic material the viruses contain may be DNA or RNA, which can either be single or double-stranded, and they can be linear or circular. Viruses that have a single strand of RNA as their genetic material might have positive sense RNA. That is, it can be immediately used in translation as messenger RNA by the host cell's ribosomes to start assembling viral proteins. Or the virus might have negative sense RNA, which is complementary to viral mRNA and must be first complementary base paired by RNA polymerase to form positive sense RNA before it can be translated at a ribosome. Or, in the case of a retrovirus, the viral RNA is used to make double-stranded DNA, then uses the negative sense strand of the DNA to make a positive sense strand of RNA that can be used in translation. Bacteriophage lambda that infects E. coli can follow either the lytic or lysogenic cycle to replicate. Regulatory genes determine if DNA is immediately expressed and follows the lytic cycle, or integrated into the host genome in the lysogenic cycle. Healthy, actively dividing cells favor the lytic cycle, and stressed cells favor the lysogenic cycle. In the lytic cycle, the bacteriophage, or phage, attaches to the surface of the host cell via the proteins it contains in its tail, which bind to maltodextrin, a protein the bacterium uses for absorbing carbohydrates. Bacteriophage lambda has double-stranded DNA that it injects inside. The host cell's enzymes and ribosomes are then used to make new copies of the viral DNA. The viral DNA is then used to make messenger RNA, which is translated to synthesize viral proteins. These components assemble into complete virus particles inside the cell. When the cell is full of new viruses, it bursts open, or lyses, releasing them to infect other cells. This process destroys the host cell and quickly spreads the infection. The lysogenic cycle begins in the same way, but takes a different course. After entry, the viral DNA becomes integrated into the host's own DNA to form one unit called the prophage. The prophage can remain inactive for many generations as the host cell divides, copying the viral DNA along with its own and passing it down to daughter cells. Under certain conditions, such as stress or damage to the host cell, the viral DNA becomes active again, separates from the host genome, and enters the lytic cycle, and begins replicating its genetic material to produce new virus particles that will exit the cell. Although viruses have many similarities, they also display a wide range of differences, as illustrated by these three examples. For instance, the bacteriophage lambda infects bacteria E. coli and has a non-enveloped capsid with double-stranded DNA. It can infect its host cell by either a lytic or lysogenic cycle. COVID-19 is a coronavirus, meaning it has a crown-like shape. COVID-19 is enveloped by a membrane with proteins embedded in it. The spike protein binds to receptors on host cells, which allow it to enter through endocytosis. It infects animal cells with its single-stranded positive sense RNA. HIV is a retrovirus and uses reverse transcriptase, an enzyme that will copy its single-stranded RNA into double-stranded DNA that it can then insert into the host cell DNA. HIV targets the helper T cells in the human immune system, which disables the system that functions to protect humans against viruses. The question of how viruses originated does not have a single answer. Scientists have proposed several hypotheses. The diverse nature of viruses suggests that they evolved independently several times throughout history, rather than all branching from a common viral ancestor. Because viruses are obligate parasites, cells must have evolved first. For viruses to have evolved, some of their similarity to cell structure suggests that they evolved from cells. Given the assumption, there are two main theories. The first is the progressive hypothesis, which suggests that viruses evolved from fragments of genetic material that escaped from living cells and became independent, also known as the escaped genes theory. This is evidenced by viruses having retrotransposons. Retrotransposons are DNA sequences found in eukaryotic cells. These sequences will synthesize a complementary messenger RNA from its sequence. The retrotransposon sequence also often contains a sequence to synthesize reverse transcriptase, an enzyme that can make DNA fragments complementary to RNA. The RNA is reverse transcribed to complementary DNA, which is then inserted into a new location in the genome. A reasonable conclusion is that if a retrotransposon sequence were to escape a cell, it could become a retrovirus. Another theory is the regressive hypothesis, which suggests that viruses evolved from small parasitic cells that gradually lost genes as they became more dependent on their hosts. This theory is evidenced by a group of bacteria that are much smaller and parasitic. They replicate within a host cell, often using their metabolism, much like a virus would. Over time, these parasitic bacteria could become more reliant on their host cell's metabolism and lose functions like protein and ATP synthesis. The diversity among viruses suggests that multiple origins may have given rise to various viral groups with multiple common ancestors, and their similarities suggest convergent evolution. Recall that convergent evolution is when species of different origins share common analogous features because it solves a common evolutionary problem, like the insect wing and bat wing that evolved separately. It is also believed that viruses have analogous structures from different lines of evolution that solves similar problems. For instance, viruses have similar capsid shapes despite being from different viral families, as some families have DNA genomes and others have RNA genomes. Despite their different common ancestors, both families have developed icosahedral capsids that are efficient geometric shapes that can self-assemble to enclose genetic material. Viruses also show analogous envelope proteins like the RNA virus influenza and the DNA herpes virus that both have glycoprotein spikes that attach to host cell receptors, despite them having different common ancestors. Finally, many viruses have evolved fusion proteins or receptor binding proteins that enable entry into the host cell by similar mechanisms despite their different ancestry. Viruses evolve at a rapid pace because they have short replication cycles, high mutation rates, and strong selection pressures driving natural selection. Evolution occurs over generations and relies on genetic variation, which can be introduced by mutations. Because a new generation of viruses can be produced in less than an hour, this speeds up the rate of evolution. Mutation rate is high in viruses, as they are often using host cell enzymes to copy their genetic material that are more prone to errors. As a result, there's a high variation for natural selection to select for or against. The selection pressures that drive evolution through natural selection are often quite high, as organisms have defense mechanisms that directly target and select out viruses that are weak to their defenses. Antibodies in the immune system can directly target antigen proteins present on the surface of the virus. If there is a mutation, the shape of the antigen's protein can change, and it's no longer complementary to the antibody. The virus with the mutated antigen evades the immune system defenses and reproduces rapidly, while the virus with an unmutated antigen can be quickly destroyed by the immune system, preventing its replication. The unmutated virus is selected out of the population, while the mutated virus replicates rapidly, producing the next generation. This is how new strains of viruses can evolve relatively quickly. The influenza virus is an enveloped virus that contains negative sense single-stranded RNA that is broken up into 8 different strands. To replicate its RNA, it uses the enzyme RNA replicase, which does not check for errors, therefore leading to a high rate of mutation. The influenza virus also increases its genetic variation by mixing genetic segments from different strains. If 2 viral strains infect a host cell at once, the RNA strands can mix in the next generation, producing new combinations that emerge rapidly that can spread widely. Another example is HIV, which, as we saw earlier, is an enveloped retrovirus. It uses an enzyme called reverse transcriptase to copy its RNA into DNA. This enzyme also does not check for errors, resulting in high mutation rates. HIV also targets cells that have an enzyme called cytidine deaminase that changes cytosine to uracil. This enzyme causes even more mutations in HIV, causing HIV to have the highest known mutation rates in viruses. A person infected with HIV can develop different strains within their body due to this high mutation rate, which can help it evade the immune system, evolve antigen proteins to invade new cell types, as well as quickly evolve to resist antiviral treatments. This ability to evolve rapidly helps a virus adapt to new hosts and survive changing environments. It also presents major challenges for controlling viral diseases and developing long-lasting vaccines. In this video, we covered virus structure, diversity, reproduction, and evolution. Below are some of the key concepts summarized from this video.